Okay.
So this is in the slide. This is like a pointer. Yeah. So there you go. Is there a microphone? Yeah, if you'd like to. Oh.
Okay, ladies and gentlemen, we're going to get started here with our next presentation with CytoDyn, company we are looking at coverage for, biotech. We're again rallying down the stretch with some of, I think, our stronger plays, and a little bit about CytoDyn. Presentation will be given by Dr. Nader Pourhassan. And CytoDyn, headquartered in Vancouver, Washington. And they're a clinical stage biotech company focused on commercialization of humanized monoclonal antibodies for the treatment and prevention of HIV. Very important. So without further delay, Dr. Nader.
Thank you. And thank you for everybody being here. So CytoDyn, in CytoDyn, the lead product is Pro-140. It's a first self-injectable antibody in late stage. Late stage means past phase 3, ready for BLA submission, which is a final approval process. It's also in early stage development for cancer indications and other immunologic disorders. Early stage here means some of the indications are in phase 2, so not all of it are early stage. So I will be hopefully figuring out how to, this is not working.
There we go.
Okay. Great, thank you. So I will be covering some material which requires forward-looking statements. I won't be reading this. Okay, our Pro-140 name provisionally is leronlimab. It's a humanized monoclonal antibody. Now, the first monoclonal antibody was developed by Kohler and Milstein in 1975, who were awarded Nobel Peace Prize later on in 1984. Monoclonal antibody have selective specificity, outstanding safety. Now, we're going to talk about the safety of our product also. Our product is Pro-140, as I said. It targets CCR5. So antibodies have to target something, bind to something, to prevent something. So we're going to talk about that. In this case, Pro-140 targets CCR5, and CCR5 is a HIV entry doorway. The person who invented Pro-140, he also discovered that HIV needs that CCR5 binding to be able to get to the cell. Just keep in mind, if HIV doesn't get to the cell, it's a common flu. It'll go away. Merck and Pfizer are testing 2 CCR5 antagonists. Both of them were developed for HIV. One of them got approval, which is Pfizer product, Maraviroc. And they're looking into their product for cancer now, their HIV product. There are more than 50 monoclonal antibodies in the market in the US currently. And the projection of 2024 is that globally, monoclonal antibodies market is going to be $136.8 billion. So with that, where is our product? Pro-140. We have 2 phase 3. One of them is already done. The phase 3 that we are talking about is done, is combination. The first one, CDO2, we call it. We finished phase 3. We had our primary endpoint with a very good p-value, 0.0032. And lately, just day before yesterday, we announced something very important. And that is, most importantly, FDA likes to see how long can the new product give a patient chance to have a suppressed viral load, which is viral load less than 50. That's the main thing. You had your primary endpoint for the first week, great, but what is the percentage? The last product approved in that area was 43%. We are at 81%. We announced that just 2 days ago. Importantly, for our product Pro 140, we have not seen any SAEs. That means serious adverse events were not reported as related to Pro 140 in this study or any study that was done before. Over 500 patients, no serious adverse event related to Pro 140. So we have met with the FDA. We announced that the FDA gave us a rolling BLA. What is rolling BLA? Rolling BLA is you can complete some of the modules of the BLA, biologic license application. And if you submit it to the FDA, they have the right to review it. But the review timeline starts till you give the last section. We believe we can finish our BLA submission by first half of 2019. We do have fast track designation, which qualifies us for accelerated approval, which is 6 months versus 10-month standard. So that's not the only study we have. The other study is also phase 3 for HIV, and it's called CD03. Now that's the exciting study that we have. This one allows patients, HIV patients, to put away all their drugs, every one of them. Some patients in the study had 4, 6 drugs. Imagine taking 6 drugs at the exact time every day. And if you miss your drug, you could get resistance. You only have 4 options. You get 3 resistance, you have a problem with your HIV, it will become AIDS. So we have 300 patient study, CD03, monotherapy, just Pro 140 alone. Patients put away all their drugs. We already enrolled, as of 2 days ago, we said we passed 303 patients were enrolled. About 40% responder rate was seen with 350 milligram, about 70% with a higher dose of 525 and increased dosing to 700. So 40% gave with 350, 70% with 525. What do you guys think we're going to give with 700? We're going to find out. Phase 2 we have in a different indication on HIV. We started doing graft versus host disease when we saw Pfizer taking the CCR5 antagonist and trying to check it for GVHD. So we have a phase 2. We have modified our protocol recently, and the first 10 patients in an open label, once we get the results, we're going to do an interim analysis. And then we're going to look at new broader focus with Pro 140 to include cancer and other immunologic disorder. In that regard, we are doing acquisition, and we are in process of that, and we will be talking about that because we have promising preclinical results in breast cancer and great preclinical, which is animal study actually, done in colon cancer and MS. Oops. So what is the mechanism of action of this product that does such a good job? So there is 2 things about monoclonal antibody, epitope and affinity. Epitope is where it binds. Affinity is how strongly can it bind. Will it fall off after it binding? So this is a HIV picture of an HIV right here. In here, you can see the smaller picture where it goes to the cell, uses the copy machine, and copies 1 billion copy in 24 hours. So when you want to stop this from going in, the founder of this product says, let's find something to bind here. Why? Because this HIV needs to bind here and here before the membrane can open up and fusion happens. So to block HIV entry receptor CCR5 is what we do right here. However, there is something else happening. CCL5 also needs to bind to CCR5 for inflammation purposes. That's what CCR5 is good for. And this interaction is also going to be disrupted by this process, which is a very good thing that we've been finding out more and more about it. And that's why we found out that cancer, that's why Pfizer found out from their product that they have indication in leukemia cancer patients who needed bone marrow transplant and problems with GVHD. So why do we think Pro 140 is such a great product? product for HIV. If anybody tells you that we are developing HIV and we're going to make money out of it, you should leave. Gilead has done a great job, spectacular job to be specific. We've done a spectacular job. This product was sold to us because there was 15 more years of development, and the founder of the product, who was CEO of Progenics, said that we're going to focus on cancer product that was very successful for them and sell this one. And after he stepped down, he became our chief science person and science officer, and he has led this all these great things that we have in the last 4 years by helping me a tremendous amount. Currently, if an HIV patient passes HIV to somebody else, that means they have a viral load more than 40 copies in 1 milliliter. Usually they have millions of copies, and that becomes AIDS. So if their viral load is less than 40 or now even 50, the transmission rate is 0. In some states, it's crime for an HIV patient not to tell their partner they're HIV. When this problem, this solution of undetectable viral load, you cannot transmit HIV, now they're looking at the law in some of the states. Maybe they change that. If the patient has complete suppressed viral load for the last year and they're checking it every quarter, maybe they shouldn't make it a crime. But Anthony Fauci, the head of HIV, said that there is only 35% of the patients have suppressed viral load. We just talked about Gilead having spectacular product, right? Why is there 35% of HIV patients suppress viral load? Why not 100%? They are a spectacular product. You take 1 pill a day if you're HIV and you're good. All the research showed that usually compliance is 65 to 67%. And they're going to miss their pill. They're going to go to a trip or something and they forget to get their pills refilled. And that is when Pro 140 becomes perhaps very important. One of the things that it has beside that is a higher quality of life. Now, in regard to 35%, if the quality of life is not there, what happens? The patients start missing their pills. It's going to say, give my body a break. Pro 140, if they take injection of Pro 140 with their product, they have 3 days blanket. They could miss their pills by 3 days. If you're on 6 pills a day and you got a chance to miss your pills for 3 days, you won't be paranoid. But if you can't miss it by 1 hour, you will be paranoid for the rest of your life that you're taking these pills. So quality of life contributes to that 35% number that you guys see up there. Now, Pro 140 has shown it has hardly any side effect or toxicity because it's an IgG4 antibody, and IgG4 antibodies are known for not having this kind of problem, side effect and toxicity. Every product out there has that is approved for HIV. Better compliance. Perhaps if they just take 1 sub-Q dose, which is 2 shots in our case, one after another, like a diabetes people taking that every day, once a week, 2 minutes a week, that might give them much better compliance rate. I think they would. And in the case of combination therapy, it gives them coverage. So to summarize, the advantage of Pro 140 side effect, over 400 patients, and now it's about 500 patients. In 8 different clinical trials, 0 SAEs related to Pro 140. The side effect on the labels of the pills go from diarrhea, nausea, I mean, you name it, it's there almost. Negligible toxicity. The toxicity for this product from short to long term. When you take a 500 milligram safe synthetic chemical product, I don't care which one, just take any of 500 milligram. Take it twice a day and take it for 10, 20, 30 years. It's going to hurt the organs. That problem is not there with the Pro 140. The resistance, no drug resistance in patients on monotherapy for over 3 years. When I say over 3 years, I'm talking about close to 4 years now. Patient taking nothing but Pro 140. You take any drug, and they will build resistance if you give it by itself. There's 76% of HIV patients have at least 1 resistance right now. That means there are 4 options is now down to 3 options. If it goes down to 1 option, that's it. They don't have viable, highly active antiretroviral therapy. So That's in regard to resistance. Our product could not build resistance. Why? Why didn't it build resistance? Well, if the resistance happens because the copy that the virus makes of itself and it has a sloppy copy, if it can't go in the cell, then it can't copy itself. So there's no resistance. There's no copy. There's no resistance copy coming out. There's no copies coming out. How about compliance? Weekly, easy, subcutaneous, self-administer at home. Isn't that better than going to hospital every month or every 2 weeks? The other products that are trying to do the same thing. So daily lifestyle dosing of current drugs that are approved have only 35% of patients in the United States with suppressed viral load. Dr. Anthony Fauci said that's not acceptable. I agree with him. Okay, so let's go over our combination therapy real quick. There was 50 patient protocol. We did 52. These patients are R5. If they're not R5, our product doesn't work on them. Early stage HIV patients of almost 85% are R5. And they were failing their heart. And while they were failing their heart, they already had 3-class resistance or 2-class resistance with limited options. So we had a hard time enrolling this because you have to sit in the hospitals, and as soon as the patient checks for their viral load, which they do every 6 months or every 3 months, then if they fail, then you check them for this trial. We had our primary endpoint. A lengthy enrollment of 52 was done, and primary endpoint was hit. All of these patients in our trial, 50% were going with Pro-141 shot and 50% with placebo for 1 week. That's what our primary endpoint was evaluated at. And the Pro-140 patients achieved statistically significant viral load reduction with a p-value of 0.0032. Then these patients go on an open label, 24 weeks. In that 24 weeks, they get optimized background regimen and weekly injection. Now they got 3 days blanket over their pills. That means if they miss their pills, they're covered. So in that regard, we found that 81%, latest product approved was 43%, at less than 50 copies at the end of 24 weeks. There was no report of SAE, as I said. 40 patients out of the patients who finished the trial asked for extension. They said, we don't want to get off Pro-140. We are having them in an extension right now. And regulatory path, we are expected to put our first approval for Pro-140 to FDA very soon, and full BLA should be submitted no later than first half of 2019. Then the safety portion of our clinical trial will be 150 patients. We only have 52 here, so the other patients, FDA agreed to allow us to use our monotherapy trial, which is the next trial I will be talking about. So what happened? So that was our first phase 3 combination. Now we go to the second phase 3 that we have. It's investigative. It's not pivotal. All the patients in this trial must have R5 strain and have suppressed viral load. And they said, well, I'm tired of my pills, but I have suppressed viral load. Let me just switch. This was a 48-week open label trial, investigative. And we wanted to know if we can have a higher responder rate. FDA says, first question, can you get higher responder rate? Second question, why did some people not respond? Everybody should respond, right? Why was the rate only, like, for example, 40% here? And the third question was, if they don't respond, what do they lose? They lose nothing. They don't lose any of their options in regards to their treatment. And that was a major question for FDA. Was there any harm if somebody says, I want to try this, and they're not the responders? 100% of the patients who didn't respond all went back to their original regimen. We followed some of them for 2 years. So the very, very good news that I put out day before yesterday was that with 350 milligram our original regimen, 40% responders rate. We increased it with 175 milligram, 70%. And these results, I have to say, is patients who passing 1 to 9 months, so it could vary after everybody passed a few months. Third dose is 700 milligram. Why are we going to higher and higher, and why didn't we do that at the beginning? We already had dose response curve. Because When we went to the first site and we said we're going to do sub-Q self-injection, the first question out of the mouth of the principal investigator was, how many injections? I said, how about 3 or 4? He says, no. I said, how about 2? He said, okay, that's good. That same investigator said, look, there is almost very little site injection reaction. So why don't we just change it to higher dose? We did, responders rate is a lot better. And then the same principal investigator said, you know, there's no difference between site injection reaction to these 2 doses, so let's go to a higher one. Because now, what if we get the same difference here, we get a bump on this 70%? What if it's 85%? What did FDA have for responders rate for other products that was approved? Their first product that was approved is called Atripla. You can Google it. What was their responders rate? I believe it was 71% or something. So you can see we're sitting at a very good position. Now, as I said, 100% of patients that was not responders were okay with their going back to the original regimen, no serious adverse event related. What are we going to do going forward, regulatory path? We're going to conduct a pivotal phase 3 trial after meeting with FDA and after we have results of the 700 milligram arm. Once we do that, and if the FDA allows us to do a pivotal trial, the FDA says then the results, we're going to look at it, and then if the results are good, then we will allow you to do a BLA. They said that to us already. They said we will allow you to do a BLA submission for second approval, which is label expansion. So get your first approval first with combination. And we definitely said, okay. Now I have a video that I'm not showing it here because it's difficult to show the video in this setting of webcast. That video has 9 patients who had gone 2 years without any pills. I'm just going to tell you what they said. Some of them said, where was this product 2 years ago? I'm sorry, 20 years ago. They said, I've been in HIV for 20 years. I lost my life almost because I can't go exercise. I can't go see my children. I can't go outside. I'm constantly sleeping. These products are killing me. Now, this is not the way it is now. Today's product is much, much better. I'm talking about 25, 30 years ago. They were hoping this product was available at that time. But the willing of taking this is very high, and patients talk very, very good. After 2 years of monotherapy, we took that video. Those patients are now close to 4 years into monotherapy. So what's the market size for this small indication, and what's the market size for the big indication? The small one being combination, big one being the monotherapy. Well, according to the reports out there, 2019 number of patients would be 1.421 million. So 1.4 million HIV patients in the United States alone. 775,000 will take product. To resistance, 246,842. If we say even half of these guys, or 1/4 of them, or 1/5 of them, or even 1/6 of these guys have limited treatment option and they can take our product, that's about 50,000. 50,000 times 30,000, that's $1.5 million. That's for our small population. The bigger population, if you look at this number and see how many of these would have R5, if you multiply it by 0.7, that's the number, 70%, that number goes through the roof. But conservatively, we think the combination will have something like over 1 billion and close to 4 billion for monotherapy. So that finishes HIV. Well, we're not done yet because back in 2011, when Pfizer had some patients that were taking HIV product, and after you take your HIV for 20, 30 years, you could get leukemia and then you need bone marrow transplant. And they said, wait a minute, patients who are taking Maraviroc are not having any GVHD. Maybe our product, Maraviroc, is good for GVHD. Why didn't Pfizer know that already? Why wasn't that information already in the public domain? Because CCR5 and the CCR5 role in inflammation was not defined. It's still not defined 100%. There's a lot of research has been done in the last 10 years. When Pfizer found out about this, we immediately did a study of Pro-140 in vitro, and it was positive. We sent the in vitro, not even animal study, in vitro study to FDA, and they gave us phase 2, because this is a deadly disease. So we believe, among other potential GVHD, there is autoimmune disease disorder and cancer, including tumor metastasis, and we will talk about that in a minute. So when we got phase 2 for GVHD, we immediately said, let's file for orphan drug designation. You know, that sounds good, and maybe, you know, we can get it. FDA said, well, you don't have any animal study or human study. Why don't you wait till you get human study or do this animal study? This one, they said, take two sets of mice, and one of them, both of them should have human bone marrow. Inject human bone marrow, so they now have a new immune system. It's called humanized mice. Big pharmas would love to have humanized mice. You know why? Because they can check their product whether it works or not. And if it's a humanized mice, that would be a great thing. So you could sell that product very easily if you have a humanized mice. If you get it, you got to patent it right away. So we gave these mice A and B human bone marrow. The one in A, their body weight went down, 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 and they all died. The one in the right, B, we gave Pro 140. The body weight went up, 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 and nobody died. We said, okay, there is something wrong here. We made a mistake. Let's do it again. Usually in this kind of experiments, we did it again, same results. We published this data in scientific peer review, and we have got very nice feedback from all the scientists in the world about this. We submitted that to FDA, and the FDA gave us orphan drug designation. So we modified our protocol. It's a 60 patients protocol, but the next 10 patients, when we see any results, we're going to do interim analysis, and obviously we have orphan drug designation. If it's a deadly disease with 10 patients, we're going to ask FDA perhaps for breakthrough designation if the results are there. And in regards to other indications, we have named, acquisition that we just talked about. And the person who is called Dr. Richard Pastel is a world-renowned oncologist. Dr. Richard Pastel is going to be our chief medical officer once we finish the acquisition, and we are taking his advice. He came to us. He said, I work on a product from Merck and Pfizer. They are CCR5 antagonists, and the results are great. But those products have side effects. People on chemotherapy can have side effects and toxicity. Let me work on your product. We said, please. The results came, in vitro results were very promising. So he immediately agreed to have this acquisition, come and work with us as a chief medical officer and head all of our studies. His company is called Prostagene. He founded Prostagene, and he has done some nice research, showed 50% with breast cancer had overexpressed CCR5. What does that mean? Well, if the cancer tumor has a CCR5 on it, that means those cells that have CCR5, they're the one that metastasize. That means they go to another location, and that's what kills people. If a cancer patient does not metastasize, the rate of death goes way, way down. So he thought this was so important for the world of cancer that he said, let's have a meeting. So I met with him several times, and he emphasized, you don't know what you have. Your product is great for HIV, but this could be incredible if our animal study that he wants to do comes out as strong as he thinks it's going to come out. From in vitro study, he thinks it's going to be great. So he said the cells that have CCR5, if you bind to CCR5, if he has indications, if you have indication in cancer, we should explore that immediately, and we have agreed to do that. So we're going to have an expanded role for Pro 140 in cancer. Our focus is going to be not away from HIV, but it will be also adding cancer to this. We have a letter of intent to acquire Prostagene. That due diligence is going on right now. As soon as we're done with that, we will be going full force and full velocity going forward. And I believe that we can file IND to initiate trials also for colon cancer. That study, Dr. Pastel wasn't involved in that one. We have got the independent study that was done with colon cancer with Pro 140. The results were spectacular. So we will definitely explore that, and we also might initiate an IND, initial new drug application for phase 2 in multiple sclerosis. So this is the last one. I'm going to tell you just quickly what happened. Our chief scientific officer, Dr. Dennis Berger, took this product to Providence and in Oregon, and he gave it to the people who knew to test it for multiple sclerosis. So 6 mice were used. 3 of them got Pro-140, the other 3 didn't. They were all exposed to MS. The 3 that had MS without Pro-140 had paralysis cycle. The other 3 didn't have anything, 0. They were shocked. They said, where did you get this product? And we are going forward with them on that. So anything that has to do with CCR5 in these diseases that we're talking about, we will check. So our milestone going forward, obviously, we're very, very proud that we hit a primary endpoint. That's a big milestone for any company. Medical conferences, presentation, we had done that in CROI, ASM. In CROI, we were selected as team discussion among all the attendees. 5 were selected, 3 was from Gilead, 1 from Merck, and 1 was us. We have published studies in graft versus host disease, preclinical study that's done. Orphan drug designation was given to us. Publication from our first monotherapy phase 2 is already done. Publication for combination therapy trial will be done in second half of this year, which is anytime now. And CDO2, which is the combination pivotal trial, we're going to be we presented it at ASM Micro as a late breaker. And BLA submission first half of 2019, and file for HIV breakthrough designation will happen this year. And IND submission for 2 indications for phase 2, colon cancer and MS, will also happen this year. So with that, I go to question and answer, please.
Yeah.
Go ahead.
The indications in addition to HIV, do you have some type of a long-term feeling for what they could be in terms of the big picture, what they might bring?
So you're asking, beside HIV, the other indications, what they could bring for CytoDyn, correct? According to Dr. Richard Pastel, he believes that the impact on the patient population would be incredible if we have positive results. We don't have positive results. We don't want to get ahead of ourselves. But the fact that other products that are CCR5 antagonists had success, that's encouraging. And we are just going to explore. One quick note is we took 4 years to take Pro-140 from phase 2A to end of phase 3. Usually it takes 15 years, 10 years, 15 years. So we're going to use the same speed going forward, especially with Dr. Richard Pastel as our new chief medical officer.
You took the HIV on the orphan path? Is that how you read it?
No, we asked for orphan drug designation for the combination therapy. And the FDA says, the letter says, denied because your monotherapy could sell for more than 200,000 patients. So you're not going to be selling to a small population. You're going to sell to a very large population. So orphan drug was denied, but we were pleasantly happy to see that denial. Yes?
So what is the correlation in the mechanism between the HIV and the oncology indication? Like what's the sort of hypothesis in terms of why it's the same mechanism that works for both? I've worked in both,
Sure.
in both of those categories. So
Yeah.
just curious.
So that question is not answered yet. We are still doing studies, but I can tell you just briefly what it is. So the CCR5 binding that happens is exactly the location HIV uses. So that's clear. That's very easy to understand. But downstream, when there is inflammation in the body, the CCL5 works like a GPS. It takes the cells to the location of inflammation. That binding that happens for us with the CCR5, it interacts with CCL5 binding also in some ways, not in the full amount, because if it does in full amount, the chemokine binding will be stopped. That's not stopped. We bind in such a way where that is not stopped, the chemokine binding, but again, it has implication in cancer and everything. We were not looking for cancer or GVHD because of that fact that we don't interfere with chemokine binding. But as we saw Pfizer file for GVHD, we checked it and it had indication in vitro. Then we did a mouse study. So that answer, that question that you're asking is very complicated. We like to get the answer fully answered, the question fully answered. So hopefully we have that very soon. We are doing a study that the results should be out any week, and that has answer for that, exact answer for that. Okay, anybody else?
Regarding the time frame to commercialization of the HIV Pro 140, how elongated is that time frame, and have you looked at potential marketing distribution partners yet?
So your question is for commercialization of HIV, have you looked at other partners and how that process works? Yes, we have. We have announced already that we are pursuing all the possible partnership and license agreement opportunities. We cannot talk about any one of them because of the confidentiality. But let's just assume nobody buys us for HIV. HIV is a very unique therapy that only certain hospitals and certain doctors sell that product. Any product you take, you know how to sell it. That's just buying and selling. There's regulations here. You just got to follow them. There are sales reps right now selling HIV product. We can just hire those sales reps and just sell our own product. Making product in a very high level, that means making product for $1 billion worth of sales, that's a huge undertaking. We like to have partnership, but if we don't, we will not stop. We will just continue. Anybody else? Well, I want to thank you everybody for. Oh, I'm sorry. Go ahead.
The new 700 milligram requirement, is that just for new patients or the ones that didn't respond to the 525?
That's a great question. So you're asking. I'm repeating the question, so it could be. So you're asking the 525 that we use and the 700. Is this for new patients or patients who failed the lower dose? The 350 milligram patients. Once we incorporated the 525, we had a randomized situation where the FDA says randomized between lower and higher dose, so you can see exactly the effect side by side. But if anybody fails the lower dose from before or on that randomized division, you can have them, if they select to go to higher dose, you can do that. And just in our two arms that we have, when we had lower dose failing, when we put patients on higher dose, the viral load went right back to suppression on most of them. Most of them, maybe I would say 80, 90% went right back to suppression. That was a great indication that, hey, we're not at the higher dose. We need to go higher. And now that the principal investigators told us, let's go to even a higher dose because you can stay with 2 injections, just change the volume. 1 milliliter went to 1.5, and now we're going to go to 2. So, yeah, go ahead.
So it seems as though there's a mechanism that reduces the level of resistance in HIV, correct? And that might also be applicable to oncology. That's a big issue in both diseases.
So you're asking if this product will reduce resistance in HIV, which could then be applicable to other indications. The resistance in other products that patients have, let's just say patients have resistance to class 1 and class 2. If they take our product, that resistance will not go away. The resistance, the evolution, if the patient doesn't take anything, you will feel like the resistance will go back to the original wild virus, so it will revert back. But that's something you have to do huge studies and find out. But we don't have resistance to Pro 140. Okay, yeah, to our drug, because we don't have any copies coming out of the cell. We coat the cell, so no copies, whether it's resistance or the original replication of the virus does not happen. So does that have anything to do with non-HIV? I don't know if I can answer that at this time till we get more results. I don't know. Go ahead. Go ahead.
The drug has to be refrigerated?
Yes. Yeah,
so it
definitely.
So it would be the traditional hypodermic needle sort of thing?
That's right. And you got to put it in, room temperature for 24 hours or a little bit more is okay. Our stability data are very strong. Every product that we make, we put them on stability. Some of them are on stability for a couple of years. We had product from before, which was in negative 80 degrees Celsius, frozen drug product, change it to drug substance. And those, they were fine. There was no problem with the stability binding side. Stability was great. So we have seen no problems with the product. Any other question? Okay, I want to thank everybody for being here. Appreciate it.